manufacturing of the HEV-SG vaccine for the Nipah virus disease. The Nipah virus is an emerging zoonotic paramyxovirus. It was first detected in Malaysia in 1998 and has presented several outbreaks in much of Southeast Asia and South Asia. The RNA genome consists of a consecutive arrangement of 6 genes, including the nucleocapsid, which is the N, the phosphoprotein P, matrix fusion glycoprotein, and the attachment glycoprotein. The attachment and the fusion glycoproteins are the ones that attaches to the cells and starts the infection. It is transmitted via inhalation, and it can be ingested through an infected material. And most case fatality rates usually end up to be 100%. So currently, there are no drugs or vaccine that targets Nipah virus in reality. And because of that, it is identified as a priority disease for the WHO. It is also in the Coalition for Epidemic Preparedness Innovations priority list for similar reasons. So there are 3 vaccines that we have. One is the live recombinant RAV vaccine, which uses the rabies virus. So it's a virus vector vaccine. Similarly, there's also another one that is the RVSV Nipah virus vaccine, which is also a viral vector vaccine. The big disadvantage of this is since we are using a viral vector, it tends to be less safe compared to the recombinant subunit HEV-SG glycoprotein. Because of the safety in mind, we decided to choose the recombinant subunit glycoprotein vaccine. So because it's a subunit vaccine, there is a big downside, which is the efficacy of it, but we can counteract that with an adjuvant. Currently, the HEV-SG vaccine is in clinical development, but it is also used as an emergency countermeasure for NIV in case of an outbreak, which is also another reason why we picked this vaccine. And for the manufacturing of the vaccine, we decided on 4 main factors of manufacturing, which are the cell culture, the purification of the cells, the isolation, and the formulation of the vaccines.
So now I will talk to you about each step of the manufacturing process separately. So the first important step is cell culture, and for our vaccine we decided the cell culture to be the heterologous expression system for the vaccine to be human embryonic kidney cells, and in this figure we can see the sample HEK293 model. So we also tried to consider bacterial cells, but due to the fact that they are not able to fold proper proteins, we decided not to choose that one. And in cell culture, it is one of the most important factors to determine an appropriate design of a bioreactor in which the cell growth happens. So the bioreactor affects the yield and efficiency of the cell culture, and it's also affecting in its turn the final product efficiency and quality. So considering this fact, and also due to the fact that perfusion bioreactors provide cell density and volumetric production rate up to around 100 times higher than the batch reactors, we decided to take perfusion bioreactors for the cell culture. And in cell culture, one of the most important factors is also the optimal state of the cell line, and it's important to keep the cell line in the optimal state for the efficiency and quality of the final product. And this can be achieved by by controlling several factors, including physical and chemical parameters. So in this regard, some strategies can be done, including pH control, temperature, or pressure control strategies. Next slide, please. So one of the important steps is also the isolation of the antigen from the basically separation of the antigen from the host environment, which is usually done by cell lysis method. And it is important to make sure that all impurities are removed before moving to the next steps. And for the cell lysis, usually mechanical and chemical lysis methods are applied. In mechanical methods, we can mention high pressure homogenizer or feed mill methods. In these methods, in high pressure homogenizer method, high pressure is applied and the cells are disrupted by the shear force. And in bead mill method, the agitation of tiny beads is applied to disrupt the cells, and these beads are usually made of glass or ceramic. It's also important to note that the high rate of heat generation in the high pressure homogenizer method can be a problem, but this is usually minimized, controlled by applying some cooling systems. And it's also the fact that it is difficult to purify the light samples by mechanical methods, the samples which have been lysed by mechanical methods. And there are also chemical lysis methods, especially alkaline lysis, which is more suitable for the isolation of sensible intracellular components, especially proteins. And in these methods, OH ions are used to disrupt the cells. But considering that during an outbreak, there is an increasing demand for the vaccine, and also considering that mechanical lysis methods provide higher efficiency and higher throughput, we decided to use mechanical cell lysis methods for the isolation of the antigen in our project. Next one, please. So after isolation, the purification of the target protein should be done, which is the separation of the target protein from the impurities of the host cell, including nucleic acids, proteins, and lipids. And it is also important to apply such a method that will keep consistency for the production of final product in a commercial scale. So the purification of these types of vaccines, the subunit vaccines, is usually carried out in 3 steps: capturing, intermediate purification, and polishing. So capturing is basically the first step, the initial step, the separation of protein from the crude solution. And this step makes the product more concentrated, and it can be carried out by applying ion exchange adsorbents. And in the intermediate purification method, purification step, different techniques can be applied based on the resolution and the recovery level, such as hydrophobic interactions and ionic exchangers. And we decided to anion anion exchange columns for the absorption of proteins in this step. And the last step is polishing. In this step, the minor impurities are removed, such as nucleic acids and endotoxins. And considering the smaller volume and concentration in the last step, we can apply size exclusion chromatography in the last step. Next one, please.
So after all the purification is done, we can start making the final vaccine for use, which is the formulation step. This usually includes the adjuvant and the preservative. So the adjuvant, it's an ingredient that elicits a stronger immune response to the protein, and the ingredient can usually reduce the amount of protein needed for a vaccine by dosage. And for example, the NF59, it's an adjuvant that can reduce the dosage of like the flu by roughly 100 times. However, it is a oil and water emulsion adjuvant, so it doesn't really store well. Instead, we decided to use the aluminum salts adjuvant, which is aka Alum. It's used for this vaccine due to its safety. It's also easy to produce, and it's very flexible for other diseases. Finally, we need to choose a preservative, so we decided to use the thimerosal preservative. It prevents bacterial growth, and it prevents clumping in the vaccine, and the side effect of that is usually just swelling and redness. That's also a similar side effect for the Ellume adjuvants. So we had to decided to pick an ingredient for the SNS or a component for the NS, and we decided to choose the adjuvant, and we considered the ease of storage, ability to meet the increased demand due to an outbreak, and the comparison to the other components. So for the cell culture, isolation, and purification, they all had a very similar problem, which is the overload of downstream processes. So if you improve one of these components, such as like making more cells from the cell culture, maybe the isolation can get overloaded, or they can't separate as many cells, and this can lead to overheating and denaturing of the protein. And let's say if we actually did have proper isolation, there might be too much protein for the chromatography to separate, and that could clog the chromatography column. And also, if we actually had enough protein, these are also just subunits proteins, so they are not effective without the adjuvant. So if we didn't have enough adjuvant, then these subunit proteins are not effective for a vaccine. And even if if all three components were, let's say, operating at a capacity just to prepare for the outbreak, so they're operating at a lower capacity in case they need to accelerate their capacity, companies will not probably be less willing to agree to this because they are incurring a opportunity costs for operating at a lower capacity. So we decided to choose the adjuvant because reducing the dosage, or the ability to reduce the dosage, means less stress on the other processes. And it can also just mean that, like from the previous slide, adjuvants can reduce dosage by 100 times, or the amount of proteins by 100 times. That's kind of like similar to saying that you can reduce the amount of proteins you need to produce, or the amount of proteins or effort that you need to produce by like 100 times. So that's a lot less production that you need. Additionally, Ellume is also non-perishable in its salt form. So as long as it's not in a solution, you can store it in room temperature, making storage really easy. And finally, Ellume isn't just confined to the Nipah virus vaccine. It can be used for other vaccines. So in case there's another emergency, such as like something like COVID or a disease similar to COVID, you can possibly use the aluminum salts adjuvant to make vaccines for those outbreaks as well. And that's it.